But this is an impromptu panel. We've integrated it into the agenda, and it's so exciting that these experts are here to generously provide more time to us to provide an interactive panel on research, Q&A, and also how that also dovetails into clinical trials. So we have Dr. Mike Heinrich from OHSU, Dr. Samantha Greenberg from UT Southwestern, Dr. Blakely from the NIH. And if Jim Hughes is around, we'll have Jim also join the panel as well, because he's our clinical trials coordinator. So we're going to keep this very open and interactive. And I don't know if there's one who wants to get started on just doing a startup of the slides that are up here. Okay, thank you, Dr. Blakely.
Sure. Thank you for the opportunity. So I'm just going to run through this very, very quickly. And this is my natural history study at the NIH. And we will say that everybody's on a clinical trial, but it's really just a clinical protocol. And so there are natural history studies and there are interventional trials. This is fundamentally a natural history study because we're not doing experimental surgery, but we are trying to explore the impact of surgery in patients who have GIST. So this trial opened about 3.5 years ago. We have 65 patients enrolled on the study. We've done about 45 surgeries on the study. And then in November of last year, we started the upper endoscopy as a component of every surgery that we do on the trial. Part of this for clinical indications, because a lot of patients can benefit from an upper endoscopy, even if it's a gastric or small bowel primary, but it also facilitates capture of research specimens, and this feeds into our studies of the microbiome. And I think yesterday there was a session on the microbiome and nutrition and its interaction in GIST development and progression, I think is an open question, and that's what we're trying to explore as completely as possible. The other part of the trial is that when we harvest this tissue, we prepare it in various ways for biobanking and future use. And so a lot of what we end up hearing about mostly is formalin fixed, paraffin embedded. That's what pathology will tend to do with specimens, but what we also do at the time of surgery is take pieces of the tumor and put it right into liquid nitrogen or flash freezing or snap freezing. And what we'll also do is viably freeze it so that it's cooled over about 2 to 4 hours, and that allows those cells to then be reanimated and used to make organoids or cell lines or other models of the tumor. And all of this is a component of the recently approved UO1 grant with the SDH consortium that we have with OHSU, with UCSD, UT Southwestern too? No, I don't think so. But various institutions around the country to try and centralize a lot of this, and we are serving as the biobank for that. What's unique about the NIH? As I've mentioned to some folks in side conversations, there's kind of 2 surgical approaches, and one is a technique approach and one is a disease approach. And what we are allowed to be at the NIH much better is a disease approach in surgery, and that is trying to make sure that we tailor whatever surgical intervention to be most appropriate for that patient with their specific and unique situation. There are minimal financial barriers to rendering care at the NIH. I used to say none, but basically all care we provide at the NIH, and we do the surgeries at the NIH. I've had patients ask me, so I'll be on the study at the NIH, and where's my surgery done? At Hopkins or where? That's a fair question, because there's even employees at the NIH who don't realize that we have a clinical center where we do surgeries, we have an ICU and full ORs, et cetera. And all of the care that's rendered on campus at the NIH is of no cost. We don't deal with insurance, we don't deal with pre-authorizations. Whatever we want to get, we can get it. The only costs are the initial travel, lodging, and food for the patient, but then subsequent visits are subsidized or paid for by the NIH. And then, of course, we have a robust research infrastructure there. The other thing that we have is a lot of multidisciplinary help, because we have experts in endocrine neoplasms for SDH-associated pheochromocytoma and paraganglioma. We have geneticists who've been working with patients with GIST for decades. And we also have researchers on the back end, of course, who help develop some of the hypermethylation assays and other platforms. So my current questions are, what are the outcomes of debulking surgery in select patients? I'm not trying to just say if you are a patient with a GIST, then you absolutely should go right to surgery. That's not what I'm trying to say. But also to say that some patients who maybe would not have traditionally been considered good surgical candidates, maybe they are. And, I think offering that as an option is good, but evaluating the outcomes of that is even better. And one thing that we want to understand is what is the significance of regional heterogeneity of the disease within patients, and also, as I mentioned, the role of the microbiome. So, with that, it's a very brief overview, but hopefully gives you a little bit more insight into the NIH and some of the logistics. There's a lot of people who go into this, and given the rather emotional award ceremony yesterday, I didn't get a good chance to thank everybody, and there's people in the room who I also want to thank for the recognition from yesterday, but there's a lot of folks who work with me at the NIH, and Audra Satterwhite was here at the conference, and a lot of you know her and have worked with her. And then our research group is growing as some of our fellows have been working with us and really trying to push these initiatives forward. So with that, I will wrap up, and then I think Dr. Heinrich has some slides he'd like to go through.
While we transition to Dr. Heinrich's slides, I also just want to say in the roundtable, a few of us were having a discussion about nutrition, lifestyle, exposure, et cetera, and how challenging it is to research some of those things because we'd have to force you to live a very rigid life and do this whole randomized control trial. So some of this microbiome and nutrition research is also the next frontier. How do we look at some of those things and see if we can't learn from the microbiome and nutrition to then extrapolate, right, what kind of dietary and other things may be beneficial long term? Good transition.
Okay, so I'll try to I have some slides, but if you have a question, since this is sort of informal, just raise your hand and maybe we'll answer your question at that point and kind of move through. So clinical trials are a little complicated in that there's many steps to them. So this is sort of a mythical timeline in which there's a patient with GIST and they're in a certain situation, so they need a third-line therapy. And so we have to kind of identify the trial, and then you have to go and be consented. So you have to travel to a site, and generally the consent form is kind of daunting because it's usually about 35 pages long because of all the disclaimers and information, of which really I think the most important parts of the consent form are the risk language, so reading what are potential side effects of what you're doing. And then the really, I think the most critical thing for most people is you look at the last 2 pages, at least at our institution, the last 2 pages are the study calendar, and so you can see what you're signing up for. So in some of our studies, you have to come weekly for the first month, which means you came a week before that to get signed up, then you're coming weekly, then you're coming every 2 weeks, and then eventually you transition to every month. And there's a lot of logistics to it before you get to the situation. And then you have to be eligible. So because we have so many people who come from a distance, we try to pre-screen you to the best of our ability. So we try to figure out what's your GIST situation, what therapies you have, what medications are you on, what do your labs look like. If your liver or your kidney function is very poor, then you probably won't be eligible. But we don't want you to fly to Portland just to be told that your kidney function is not good enough. So we try to work with you, and we also try to send you the consent form electronically so that you can think about it and talk to us by phone. Not so that when you come, you don't have to agree to do it. That's still your right to say, oh, I didn't understand it, I don't want to do it. But we want you to understand what you should be thinking about, and so that you could say, oh, well, this isn't really what I want to do. I don't want to fly up to Portland just to kind of figure that out. Usually we can figure out at a distance. And so then we get going, and there's a lot of complicated parts to that. So, we talk about the phases of a clinical study. So phase 1 is our earliest. That's usually a drug first in human. So the drug has only been given to animals previously. And so if you're at the very beginning of the studies, somebody is the first human in the world to take a drug. The goal of the patient, obviously, is to get better. But the goal of the study is to figure out how should we dose the drug. So if you think about any drug that you're on, like imatinib or Gleevec that many people in this room have been on or will be on maybe, the standard dose is 400 milligrams, but the first person who took it back in 1998 probably took 25 or 50 milligrams, and it took us a while to get up to figure out that 400 was sort of the sweet spot. So in the phase 1 study, we want the drug to work, we want it to help you, but really we're trying to figure out what is the dose and what is the safety and what are the side effects. After a drug has been studied in phase 1, we go into a phase 2 study. So a phase 2 study is we know the dose, we think we know something about the safety of the drug, and now we have a specific question. Does it work in a population? So does it work in third-line GIST? Does it work in second-line GIST? There's generally a specific question that's being asked, and at that point, probably 100 people have taken it, the drug already in the world, but that's not so many. And so we still are trying to figure out about safety. And the final phase of a study is what we call a phase 3 study. So a phase 3 study is trying to prove that one thing is better than another thing. So generally we pick what is a standard therapy and what is the new therapy, and the goal of the study is to show that the new therapy is better than the old therapy. And we generally need that type of study in order to convince the FDA to approve a drug to be used in a new way. So in phase 1, which is where a lot of GIST studies are these days, there's sort of two components. There's dose escalation and dose expansion. So in dose escalation, we're starting from the first dose that we ever use, and we're trying to go up, 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 to figure out what the dose should be and to figure out what the side effects along the way. So we try to be efficient but safe in terms of figuring that out. So we generally use something that's called a 3+3 design. And so, for example, let's say we think that the starting dose is 200 milligrams. We treat 3 people at 200 milligrams and we wait until they complete 4 weeks of therapy, and do we decide if anything severely harmful happened. We expect side effects, and if they're mild, we're fine. If nobody has a significant, what we call a dose-limiting toxicity, which means a really bad side effect, we go to 300 milligrams and we do the same thing again. And if 300 milligrams is fine, we go to the next dose, that's 400 milligrams. And we treat 3 people. But when we do that, we find that one of the first 3 people actually had a bad toxicity. So now we're not sure what we think about 400 milligrams. And so we treat 3 more people. If another person has a severe toxicity, we decide that 400 milligrams is not a safe dose, and we go back to the previous dose, that's 300 milligrams, and we don't try to go up on the dose. If nobody in the second group of 3 at 400 milligrams has a problem, so only 1 out of 6 people had a severe problem, then we go up to the next dose. This is all done for safety, but from the investigator and the patient perspective, this is super frustrating because we have to wait for things that we can't control. So we need to wait for the 3 people in Belgium to complete a month of drug before we get a spot that we can offer. And then if they say that we have a patient, but then they don't really enroll it, the whole thing kind of rolls out. And so when we talk to people, we're sometimes unclear when we think we could have you in our study. Like, we think we'll have a spot in 4 weeks, but if something happens somewhere else, it could be 8 weeks. Or conversely, if something happens somewhere else, it could be 4 days. And so you have to kind of be patient and be flexible. And, we try to advocate and get people in. But a lot of times, for safety, there's only 3 people in the world can get the drug. And our site might not have any of those. And so we're trying to figure out what the maximum tolerated dose. So there's pros and cons of being in the study. And so these are kind of listed. The pro is you get a new drug, you could be early on in the study. And if you think about the drugs that are approved, imatinib, sunitinib, regorafenib, ripretinib, you might have been excited to be in the phase 1 study of those drugs because you would have gotten them, 5 years before it was commercially approved. But on the other hand, you may be early in the dosage, so we pick a dose intentionally to start with that's really safe. Like we are sure that the dose is going to be safe based on our animal toxicity. But we also pick a dose, it may not be effective. So sometimes in a study, we have to get to the 4th or 5th dose before we really get into an effective range. But we've also had studies where in the first dose, people start responding because the drug works so well. So you kind of want to be in a phase 1 study if you need it. You would like to be in it in the part where the dose is effective, but that's not always so easy to predict. It also is super complicated because it has the most amount of visits, like in the first month.
So the last bullet point on your pros, if the drug is safe, patients can be on the treatment indefinitely. So there's, for phase 1, there's no like concrete endpoint.
So in all oncology intervention studies, or at least the ones we do in GIST, you are in the study indefinitely until you say, no, I'm not, I don't want to do it. Your disease progresses past a predefined endpoint that we don't think is ethical to continue, or something unsafe happens that we can't fix. But we have had, like on the ripretinib phase 1 studies, we had people on the phase 1 for 5 years, and eventually it became FDA approved, and then the study ended and everybody kind of moved over. So even though they may say the study's intended to run for 2 years, that's just an estimate of how long we think it would take to get all the results. But if it's working, you can continue it indefinitely as long as it's safe. And the other complicating factor in phase 1s is the US, there's usually only 4 to 5 sites, so you're going to have to travel, but we negotiate travel for all of our studies. So we hope to make it practical for people to be able to go wherever they need to go. Phase 2, kind of the pro is that the safety has been somewhat figured out, and now we're trying to figure out its effectiveness. But we may not know the effectiveness, and again, it might be only 4 to 5 sites, so you still are going to have to travel. Phase 3, we know a lot more about the drug. We're really trying to figure out if it's better than what we do right now, but sort of a point of contention is the placebos. Go ahead.
Is this thing working? There it is.
Yeah.
Testing. How do you work with your patients who are not in Oregon? And their, treatment, are you able to do you do telehealth, or, how do you work with their oncologist in their state? I mean, how do you build a care team there?
So currently I have licenses for the entire West Coast, so Alaska, Washington, Idaho, Oregon, and California. So if you can get to one of those 5 states, I can do televisits with you. If you got a contact from Life Raft Group about somebody, we would call you up, if you espouse an interest in one of our studies, and we try to get some information about you to see, okay, well, we have a 3rd line spot, but you're in your 4th line, so you're not even eligible. But we would try to figure things out. And at some point, though, you would have to, register with our site and be seen in a state that I have a license so that we could do kind of more a formal evaluation. All the time. And I have a slide on that. But we never want you to disenfranchise your local person, because what if you get COVID? What if you need IV fluids? What if you need a pain med? What if you have a rash? We don't want you to fly from Ohio to Oregon to look at your rash. We want your local care to go forward. But there's sort of limitations on what can be done to you if you're part of a study in terms of drugs that can be given. Okay, so, and here's kind of an example of how your exact question. So you are now in a clinical study. I am your provider. It's your first week of treatment and you have a really bad headache. So what are you going to do? You could take some Tylenol, you could take some Advil, or take this medicine you got from Amazon that says it's for headaches. So what do we think the answer is? The answer is call us. if you're on experimental medicines, there's a lot of drug-drug interactions. Some studies would say you cannot take Advil. It is forbidden. And if you take Advil, it creates all kinds of problems. So, and also, we don't want you to self-diagnose. So if something's going on, we want to know about it. Okay, so you saw me last week and you're having worsening diarrhea, but you're going to come back in 3 weeks. What should you do? Google. Don't want to bother us. We'll talk in 3 weeks. Take some of your daughter's medication. And again, the answer is call the research team because we want to know about it and we want to be able to guide our advice.
Can I make a comment about this? Just because I think one of the things that I think, and I'd love your thoughts, but I also think y'all have takes too, is to understand that these symptoms and where they get classified. So we've talked a little bit about toxicity and adverse events and some of those things, right? So there is a threshold within clinical trials that they determine what essentially is too bad of a symptom, right? That is classified then as an adverse event. They need to know about every symptom in order to be able to do that. But what I've heard, and some of you may have experienced, right, is that sometimes your perception of a bad symptom is not their perception of a severe adverse event, right? That a headache is helpful, they need to know about it, they need to be in that space, but that the threshold of what that means. And so again, I know we've talked about this in a number of sessions, but really advocating for yourself and your perception of where that symptom is and what you need to do. I've had patients come off trial for something that we consider a low-grade event, right? But for them, it's so disruptive that it's something that they don't want to continue on that route. And so these are really moments that are crucial to be in contact with the research team, for them to be able to walk you through what's going on. But also I just want to acknowledge that sometimes we use some of these big words, not big words, but we use these terms and they have really strict definitions because of how clinical trials are, but that your experience on them may differ from that, and it's imperative for the team to understand your side of that as you go through that clinical trial to make sure that they can manage that process.
A few more things. So you heard about a study, what should you do? At OHSU, you could call us, do nothing, we're not very responsive, don't bother. Or talk to your local provider about what you read about or heard about, and the answer is A and C. So you could reach out to us or certainly talk to your local provider. And, you know, we want to talk to you. These things are generally pretty specific. We need to know your mutation, we need your line of therapy, and we also kind of need to know things. Sometimes says OHSU has a study, but the lawyers haven't signed the contract yet, and so we're not really open, but we could tell you that Stanford is, and so we could redirect you. Kind of general things, keep your own records, that's very useful. We need usually to have a pathology report, and sometimes it's really hard to find in outside records, like you had surgery 12 years ago, we can't register you without the pathology report, but nobody can find it. So keep it yourself, keep your mutation report. It's good to know where your lab scans are. You know, I think the LifeRaft group has a way to do a GIST-to-RE, but you can do your own binder if you don't want to do it online. But knowing where all these things are and kind of knowing, oh, my creatinine's 3 is very useful because that determines what we can do. But kind of to the point, we still want you to have your local oncologist. We will send letters to them. If they have Epic, they'll be able to see all my long notes anyway. But if you need something locally, we want you to still have your doctor to go back to. And then how to find a clinical study. So Jim, I know you and the LifeRaft group have quite a bit of online materials for this.
Can you hear me? Oh, okay. Denise told me that this was the waiting area for the shuttle for the airport. Anybody can help me? Okay. Yeah, we do have something for you to help you. Many of you stopped by to see us and talk. We had a little demo of our LRG clinical trials database. So right now we have like 12 trials that are highlighted for GIST. Most of them are for KIT PDGFRA. But if you're looking in our database, be very careful. Look at the qualifications. More and more, they'll say KIT PDGFRA. They mean it. They don't mean PDGFRA D842V necessarily. And so you have to be careful and look and be specific to which even exon you're in. because our trials are going to be divided up based on the inclusion criteria, and those will specify, in many cases, in some cases, the specific codon, but in many cases now, the exon. So be careful when you look. Also, it's very carefully noted in our database which phase these trials are. As Dr. Heinrich pointed out, it's very important to know what you're looking at. And is this a trial for efficacy? Is it a trial to prove it in? Or is it a trial just to get the maximum tolerable dose? So we have, I think it's 12 trials right now in the United States. And if you want to find it, you can just type in justtrials.org. If you have questions about it, we have in there too links to monograms about the drugs that describe some of the work that went on in research before this was actually put into a clinical trial setting. We also have, where appropriate, reports from the conferences about the trial, the website that the pharmaceutical company has put up for patients for this particular clinical trial, and also the reports that are published in the journals. We will link those to the clinical trials, and you can look at a trial and go in and go through and look at those specific pieces of information. Also very helpful in there is the contact information. And just as a guideline, I would say, some cases we have data about each site where the listings in clinicaltrials.gov have included a telephone number or an email address for the site and the PI. That's also really helpful. But there's also a one number for overall trial entry. And you probably should call that number first. It's usually the pharmaceutical company or the sponsor. Most of these are pharmaceutical company sponsored. Some are simply the medical center sponsoring it. We just got a new trial at MD Anderson. They're going to be trialing regorafenib, and it's in a select group of patients. KIT exons, I think it's really odd. It's like 17, 18, 14, and SDH. Really an odd one. And nicotinamide has just opened that up. And there are other trials in there besides those that are for new drugs. There are adjuvant trials and neoadjuvant trials as well. And then there are just some trials to test like therapeutic drug monitoring. And they could be open for any patient. You don't have to have a particular condition except GIST and be taking imatinib. So any questions about the database or how to access it? Justtrials.org. All one word, just trials.
Yeah. And then I also call it clinicaltrials.gov. So this is the government website. So it's all clinical trials, not just any disease. And so, like for this example, what's the condition? GIST. What's another term? I'm interested in IDRX 42. So you type in IDRX 42, and then it pulls up the study, and there's only one. And then it tells you the title of the study, the NCT number, which helps identify it, and then it shows where it's running. In the U.S., they list Miami, Portland, Boston, Philadelphia, but there's 9 other locations, which they don't. Well, then you can click on the interactive map, and it would probably show you all the European sites. But then down below, it's pretty useful because it tells you all the eligibility criteria, so you want to look it up, decide, hey, do I think I fit in this? And it tells you what you get and where the phases of the study are. Then usually at the bottom, if you go down long enough, it tells you the sites that you can click on, and then usually there's a contact point. But you also, at the beginning of the very top, it says whether the study is active or closed, because some studies listed, if you type in GIST, you would see every study in the last 20 years in GIST, and most of them are completed, so you can filter that out. And you generally, there's a lot of studies would say like any advanced cancer, and so putting in cancer is not super helpful, I think, for most patients with GIST, because we have specific ideas of sdh deficiency or KIT mutations, and we want to get in that space and not get into random chemotherapy that we're going to give to any patient with any cancer, because we don't think that's particularly helpful. Any questions?
This does not follow
Right. Random thoughts on clinical trials. Or random doctors talking about clinical thought. Plus a genetic counselor.
Yeah.
I guess, so, Samantha?
Yeah.
So we talk a lot about the importance of genetic testing in different contexts. So as a practical matter, if you have a test result or if you heard your brother had a test result, how do you what's the action plan?
Find a geneticcounselor.com. So it sounds like a marketing strategy. So I mean, I think, right, we've talked a little bit about knowing mutations. I think as we've talked throughout the weekend, it's really crucial to have that information, make sure you have it on record, et cetera, especially if you're moving institutions, et cetera, making sure you are the owner of those documents. Your care team should be able to provide a number of them. If there's a genetic mutation that's been found in the family, making sure that you're set up with a care team for hereditary screening and your care. And I just want to emphasize, this is not quite the answer to the question, but I think one of the things we talk about a lot is really like order and priority, right? Your treatment and your care matters most. If there is a hereditary finding and you also need screening in that realm and your family needs it too, right? For your brother or your child, that can be their top priority to undergo screening. But in that moment, you need the treatment and to move forward in your cancer care. And if that means that screening or next steps on your actual hereditary finding waits, that's okay. We don't act like it's okay, right? Because our focus is to make sure that you get what you need. But there's also a moment that what you need is to not have 17,000 things on your plate, but to have this one thing, which is treatment on your plate, and that's okay. So in case you go to a genetic counselor or someone who says otherwise, just remember that. It's okay.
So what's the difference between screening genetic testing and confirmatory testing?
Oh, I just pre-recorded a lecture on this for our students.
Okay.
So broadly, right, the screening that's happening is really thinking about, tell me more about what, I could wax poetic about lots of things in that realm. Tell me what you're thinking.
Well, so a screening test generally includes like 50 to 70 genes for all the hereditary cancers.
Oh, I see what you're talking about.
But
Yeah.
I found out that my sister has an sdh-R46S mutation.
Got it.
Do I need screening or do I need testing for that?
Yes, you need testing for the genetic change that we found. There is some data and some research and some people out there that now suggest we should be doing screening and lots of multi-gene panels for everyone, but at the heart of it, the top thing that you need is the genetic test that your family member had, and to know that you're at a lab where the genetic change that was seen in your family member has been tested and seen at that lab, right? So that they can verify that you do or do not have that same genetic change.
So the way I think about, remember I talked yesterday about mutations of the library. So when you do a screen, you're like looking at lots of books because you're trying to figure out if any of the books have a typographical error. But if you're in a family situation, the question we ask is, in War and Peace on page 452, is the misspelled as they? And if it is, then that's your genetic thing that you have to worry about. But if it's then you have no mutation. So it's a lot simpler to look at one thing from a testing perspective than it is to order all the things.
Yep.
But sometimes it just defaults to that because no one can figure out how to do the confirmatory testing.
Correct. And if in your big test, right, as we've talked about, if you have a positive finding, right now a majority of the labs do have that free family aspect where they will go through and do the exact confirmatory testing. I think
I think our impromptu session is over. Thank you.
Thanks, everyone.
Is the bus here?